The overall goal of the following experiment is to use dietary means to alter the fatty acid composition of C. elegans in order to study fatty acid function and regulation. This is achieved by culturing the nematode food source in the presence of unsaturated fatty acids. Next, C. elegans are fed the supplemented bacteria, allowing for uptake of the fatty acid and its incorporation into the worm lipids. Then, changes in C. elegans fatty acid composition are confirmed by gas chromatography mass spectrometry analysis. Finally, after feeding on unsaturated fatty acids, the worms are scored for sterility. Results are obtained that show the physiological effects of altered fatty acid composition through development, behavior, and changes in gene expression.
This method can help answer key questions in the C. elegans development and physiology fields, because we can use both genetic manipulation and dietary contributions to alter fatty acid composition and monitor the consequences.
Generally, individuals new to this method must be careful and cautious because it is difficult to obtain consistent results due to the sensitivity of unsaturated fatty acids to oxygen and high temperatures.
To prepare 1 liter of fatty acid supplemented medium, measure out the following components into a flask. Add millipore water up to 800 milliliters and autoclave the medium along with an empty glass bottle for each fatty acid concentration to be tested, as well as appropriate graduated cylinders. While the medium is cooling at 55 degrees Celsius, prepare the working stock solution of the fatty acid sodium salt by carefully breaking open the glass vial so as not to get cloudy.